All right, so now we're in our third lecture out of 4 in our photosynthesis chapter. So our first lecture was thinking about mitochondria versus chloroplasts, looking at chloroplast anatomy, kind of thinking about the overview of the process. The last lecture was on the light reactions, and again, we'll summarize those briefly here. And then today we move on to the dark reactions, and then in our last lecture, we'll deal with regulation and then kind of some other ways to think about offshoots of the pathway. So again, light reactions, purpose of the light reactions are to generate NADPH and ATP, and ultimately water because we oxidize oxygen because we oxidize water to generate those electrons that we need ultimately for electron transport. So we'll do a brief review of light reactions, but today's focus is really on the dark reactions, more appropriately called light independent, and that's where we're going to utilize the NADPH and ATP produced in the light reactions, use them to assimilate or fix carbon dioxide into 3-carbon carbohydrate precursors. We'll actually make GAP, and then it's very easy to take GAP and make it into glucose derivatives by reversing glycolysis, and then we can make whatever types of polymers we want, starches, cellulose, whatever the plant might need. So we'll deal with that at the very end. But importantly, we're going to review concepts from our glycolysis chapter on the pentose phosphate pathway, because what we'll see is there's a reason that the Calvin cycle or these dark reactions are called the reverse or the reductive pentose phosphate pathway, because we basically use NADPH and we fix carbon dioxide rather than liberating carbon dioxide and generating NADPH. So again, we'll do a lot in our activity that allows us to draw those parallels. So again, reviewing the pentose phosphate pathway will be part of our activities. Again, thinking of the 2 main products of the pentose phosphate pathway being CO2 and NADPH that are made in stage 1. And then after stage 2, we get a mixture of xylulose 5-phosphate and ribose 5-phosphate. Again, we can siphon ribose 5-phosphate off for nucleotide synthesis, but truly if all you need really is ribose 5-phosphate, you're going to siphon up materials from this downstream part of glycolysis. So you'll siphon off G6P and go through stage 1 if you need reductive currency for biosynthesis. You'll siphon up from F6P and GAP if you need to synthesize nucleotides and make ribose 5-phosphate. But we'll review the pentose phosphate pathway thinking about those 3 stages, right? The CO2 and NADPH production, the restructuring to allow for recycling. So again, we'll spend some time looking at the details of this, looking at kind of the stoichiometry that we have for these different processes. So that'll be part of our activity to sort of review that. But the focus again is to think about how we reverse that process and utilize ATP and NADPH produced in the light reactions to assimilate CO2 into ultimately 3-carbon sugars as glyceraldehyde 3-phosphate. And then by reversing glycolysis, we can make that into glucose derivatives, which we can then further assimilate into different carbohydrate polymers. So looking at the Calvin cycle, there's only 2 stages to the Calvin cycle. So we'll spend time in our activity kind of breaking apart those 2 stages, understanding their purposes, understanding kind of what's going on. And again, the first stage really deals with the production of GAP. And the net is that we take 3 5-carbon pieces, so there's 15 carbon atoms here. Each one of those 5-carbon pieces is going to get an extra carbon by adding CO2. We get a 6-carbon piece. When we split that 6-carbon piece in half, that's how we get GAP. So we'll see that in the first 4 reactions here, and we'll see that on our next slide. But then the next stage is really recovery. We're going to siphon off one of those GAP products, and the other 5 go through a whole bunch of reshuffling to take 5 3-carbon pieces and make 3 5-carbon pieces. And we're not going to talk at all about the details of that. I would expect, though, again, if you were to look at any of these enzymes, you could rationalize why, for example, something's called an epimerase or an isomerase and so forth. But we're not going to study the details of those reactions. But again, stage 1, 4 reactions, that allows us to assimilate CO2. So we're going to have these 3 5-carbon pieces. Each one of them gets a CO2 to make 3 6 carbon pieces, which then we split apart here. So now we're going to have 3 phosphoglycerate pieces. We're going to phosphorylate those to make 1,3-bisphosphoglycerate. And again, we've seen that molecule before, so we're going to kind of think about going in reverse, going to make GAP. So rather than taking GAP and making 1,3-bisphosphoglycerate like we did in step 6 of glycolysis, we're actually going to be reversing that and going and making GAP. So lots of arrows here. Remember, the number of times you see those arrows indicate the number of molecules that are undergoing that process. So we'll spend some time looking at the stoichiometry of this reaction, understanding how we assimilate CO2, why we need ATP, how NADPH comes into play. So we'll look at the details of these reactions. Again, more so we'll spend time on stage 1 than stage 2. So again, at the end of stage 1, after those 4 reactions, we're going to have one of those GAP molecules get siphoned off and be used to make carbohydrates. The other 5 are going to be reshuffled, and that's what we see in this last part. Now again, we're not going to do too much to understand the details of these, other than I want you to know that if you've got 15 carbons as 5 3-carbon pieces, we're still going to end up with 15 carbons now as 3 5-carbon pieces. So a whole bunch of reshuffling that we see, not unlike the recycling stage that we saw in the pentose phosphate pathway. Again, we'll have a few key components that we'll look at in our activity, but not too many details in this stage 2, this recovery stage. We will spend a good bit of time talking about this enzyme that is responsible for step 2 of this process, which is called ribulose bisphosphate carboxylase, or abbreviated Rubisco. And arguably, again, this is the world's most important enzyme, because if we cannot fix carbon, we're not able to make fuels. And I say we as kind of a global population on Earth. Of course, this is not chemistry we can do. So we'll talk a little bit about this enzyme. We won't talk too much about its structure. It is a quite large protein, but we will spend a little bit of time looking at the mechanism for how this happens. And again, I'm just going to walk through this briefly. This will be part of our activity. Remember that alpha carbons, hydrogens, are acidic because we can resonance stabilize the intermediate. So this ene dilate intermediate becomes nucleophilic, so it can react with CO2. And again, reacting with CO2, we essentially get to a beta keto acid. So the reversal of this process is how we can get rid of CO2, right? Beta keto acids rapidly decarboxylate. And so we've actually added CO2 by doing the reverse of that process. And then once we have this beta keto acid, then the chemistry gets a little bit weird. This isn't not anything that is intuitively obvious, but we're going to have water here. Remember, strange things can happen in enzyme active sites. Water is going to nucleophilically attack this carbonyl and this ketone, and going through the elimination here of this group, this is how we end up getting these 2 3-carbon pieces. Or in this case, if we started with this piece, we'd end up with a 2-carbon and a 3-carbon piece. So again, 4 carbons here, adding 1 gives us 5, and once we get this beta keto acid from the addition of carbon dioxide, now the addition of water is going to allow us to cleave this labile bond and generate a 2-carbon and a 3-carbon piece. Okay, so chemistry that's a little bit weird mechanistically, but we'll take a look at that. And the reason we're going to take a look at that is in our last section, our last lecture, we're going to talk about how this enzyme Rubisco can be used to not only do this chemistry with CO2, we'll talk about how we can do it with O2. Okay, so having our electrophile here not be a CO2 molecule, but an O2 molecule. And that's some really interesting chemistry that we'll spend some time dealing with in our last lecture. So again, here's that same chemistry mapped onto the enzyme active site. So here's this 5-carbon piece. Again, we're going to have a general base that's going to serve to deprotonate this alpha carbons hydrogen, generating this ene dilate intermediate. Again, nucleophilic attack on our carbonyl here of CO2 is going to add our carbon, generating our beta keto acid. Nucleophilic attack by water, and then breakage of this carbon-carbon bond is going to generate our 3-carbon piece here, and again, protonation here will generate the component or the companion 3-carbon piece. So we generate two 3PG pieces from here. And again, here's the chemistry that we're observing. So we'll spend some time in our activity kind of looking at that. But that's kind of the purpose of what we have there. So the end of the dark reactions is going to give us a bunch of 3PG molecules. I'm not going to take time right now to summarize the light reactions. Again, this will be available for you to review, but we already looked at our light reactions in our last lecture. Kind of summarizing the dark reactions here, right? After we've finished the Calvin cycle, we've generated GAP. So F6P can be generated using Calvin cycle enzymes. We can kind of further generate that. Again, essentially reversing glycolysis. So again, further enzyme activity on GAP will generate F6P, and some new chemistry will allow us to generate G1P from F6P. And again, we're going to have what's called a phosphoglucose isomerase and then a phosphoglucomutase. These are similar to what is seen in the glycogen breakdown pathway. At the end of the day, we're going to get to G1P. And that's important because G1P is a required precursor for the synthesis of a lot of higher order or polymeric carbohydrates in plants. And so just to mention a couple of them here, when we talk about alpha amylose, remember alpha amylose is the linear homopolymer of glucose. So taking G1P and again activating it with ATP to make a good AMP leaving group here allows us to polymerize alpha amylose, generating these new alpha-1,4 glycosidic bonds. This is similar to chemistry that we saw with UDP glucose when we saw glycogen synthesis, so a good opportunity to kind of review that component from glycogen. We'll also talk a little bit about sucrose synthesis. So again, this uses UDP glucose plus F6P and generating a phosphorylated form of sucrose. Dephosphorylation yields sucrose. Sucrose is a really interesting carbohydrate in plants because it's, in being small and very water soluble, it's a transportable sugar. So again, this is the major transport sugar that allows carbohydrates to be delivered from photosynthesizing cells to non-photosynthesizing cells. And so interesting chemistry to think about there. And then the last sugar we need to think about synthesizing, obviously very important in plants, is cellulose. So what's interesting is that we've got UDP glucose again, but we have a multi-subunit plasma membrane complex that's going to be responsible for now generating a beta-1,4 glycosidic bond rather than that alpha-1,4 glycosidic bond that we see in starches. And remember, there's very different physical and chemical properties that we see between cellulose and alpha amylose in being a starch, and all that's different really is that glycosidic linkage. So a lot of opportunity to review a lot of different things in this chapter, but again, this section was on the dark reactions and ultimately the synthesis of GAP to allow us to make carbohydrates.